Have you ever been told that your all the reports, fertility reports are normal, but still you are not conceiving? The main three reports, the hormonal reports, the fallopian tube, and the male sperm count is normal, but still you as a couple are not conceiving. You have even undergone IVF, spent lakhs of rupees with good quality embryos, still have failed multiple times. The answer could well lie in a simple laboratory blood test, what you may not even have heard of, homocysteine. Yes, ladies and gentlemen, homocysteine. The high value of homocysteine is associated with recurrent pregnancy loss, multiple pregnancy failures, IVF failures, even pregnancy damage, IUFD, with severe preeclampsia, abruption. It may also be a factor of unexplained infertility and severe low counts in males. While the high value of homocysteine may just not be a laboratory marker, it is a sign of a significant deeper problem which is affecting male or female or both. But the question is, can this high value of homocysteine be prevented or be treated by lifestyle modifications, dietary modifications, and targeted supplements? I am Dr. Rajat, and over the last few decades of my infertility practice, I have seen many patients of unexplained infertility. Apparently, the reports are normal, but they are still not able to conceive. Recent reports and recent research in last few decades have shown a very important role of one, carbon compound metabolism, folate metabolism, methylation pathways, and their association with endogenous antioxidant in fertility outcome. In this video, you will know what is homocysteine, what is the relation between homocysteine and fertility. What does homocysteine actually do to cause infertility in both males and females? What are the targeted nutrient and dietary supplementation strategies to improve the homocysteine levels in the body? Watch this video till the end. You may find something very unique which you may never have heard of. You will thank me later. I am Dr. Rajat, and you started watching our YouTube channel, The Fertility Gynecologist. I started my video with a word called as unexplained infertility. What is unexplained infertility? Fertility is very simple, if I may say so. This is uterus, this is fallopian tubes, this is ovaries, this is vagina. A male and female have intercourse in the vagina. The semen gets deposited in the vagina. From vagina, the sperms, they swim upwards towards the cornu. The eggs gets ovulated. It gets picked up by the fallopian tube. From the fallopian tube, it goes inside, and sperms come from this side, and the union of sperms and eggs or fertilization takes place first of all in fallopian tube. It stays there for a couple of days, and the embryo which is formed, it comes back to the uterus, gets implanted, and that is how the fertility occurs. So for a normal fertility, you basically need three factors. The counts of male should be normal, the hormones of female should be normal, and the fallopian tube status should be normal. Apparently, in most of the cases, in approximately 90 to 95% of the cases, these three reports are normal. But still, most of them are not able to conceive, and that is what we, in our technical medical jargon, called as unexplained infertility. Yes, over the last few decades, I have researched a lot about unexplained infertility, and I found a wonderful marker, which we know since long, but it is underutilized, called as homocysteine. Homocysteine is actually amino acid which is naturally produced in body and metabolized via the methylation cycle. High level of homocysteine is associated with infertility and pregnancy complications like preterm delivery, preeclampsia, miscarriage, and low birth weight. Homocysteine is a biochemical basis for increase in oxidative stress. It is associated with poor quality oocytes, with low fertilization rate, poor quality embryos, and because of the follicular atresia, it can lead to the low AMH levels as well. Vitamin D is a very important factor in homocysteine metabolism. The homocysteine metabolism, one carbon metabolism, I have discussed in detail in my previous video regarding the active folic acid. But vitamin D is a very important cofactor in cystathione beta synthase, which is the enzyme involved in conversion from homocysteine to methionine, homocysteine to cystathione and cysteine, and then to the methionine via the transsulfuration pathway. And as a result, vitamin D plays a very important role to decrease the homocysteine level in body, and no wonder that vitamin D levels are reducing nowadays, and as a result, so is the homocysteine level which is increasing. So homocysteine and vitamin D have an inverse relationship. If homocysteine increases, vitamin D decreases, and if vitamin D increases, the homocysteine level is in check. And as I have alluded to many times before, I have referred to many times before, almost 95 to 97% of Indian adult population is deficient in vitamin D. partly because we have stopped moving out in sunlight. Vitamin D is a sunshine vitamin. It is formed in body when sunlight hits our skin, and it may be, if we remain in sunlight for at least half an hour, 20 minutes, that is the process when the vitamin D starts forming. Vitamin D level is very low nowadays because most of us like to stay in dark closed room in AC. We do not go out in sunlight most of the times, and that is why vitamin D is deficient, and that is why deficiency of vitamin D plays a role in the decrease in function of cystathione beta synthase enzyme, which ultimately decreases the level of homocysteine. It converts homocysteine to cystathione and cysteine. There is also an association between recurrent implantation failure, recurrent IVF failure, recurrent pregnancy loss, and vitamin D. People have found low levels of vitamin D, low and high levels of homocysteine in patients with recurrent pregnancy loss and recurrent implantation failure. Al-Bayari did a study and he found that vitamin D preparation of 60,000 unit international unit per week for approximately 2 months, it decreases the level of homocysteine level in the body. WHO says that at least 730 micrograms per day folic acid in active form has to be given in people who are trying to conceive. I would go on to extend to say that at least 800 microgram folic acid should be taken minimum 3 months before a couple wants to conceive, both male and female, because anybody could be suffering from MTHFR polymorphism, which is ultimately responsible for converting 5,10 methylene tetrahydrofolate to 5-methyl tetrahydrofolate, which is the active form of folic acid and which is the methyl donor for most of the protein synthesis, DNA, chromosomes, neurotransmitters, everything. This 800 microgram folic acid per day decreases homocysteine level by about 25%. The cutoff level of homocysteine which should be there in males and females should be 10, ideally 7. Maximum fertilization occurs if homocysteine level is less than 7. If homocysteine is high and patient conceives, then there is more chances of miscarriage, and if it is very high, then I would personally consider the use of low molecular weight heparin, because homocysteine is known to cause increase in thrombogenic potential. It is vasculotoxic, it decreases nitrous oxide, and if pregnancy is precious, if patient has conceived by IUI or IVF or patient is elderly, then I would consider the use of low molecular weight heparin along with aspirin to prevent the aggregation of platelets and increase in the placentation. Oxidative damage, it triggers the extensive repair in gametes and thereafter the zygote, but it results in clinically relevant damage when it affects the maturation of gametes chromatin, that is padlocking, and the epigenetic marking. It associates defective methylation and oxidation of methyl marks, leading to the derangement in the gametic epigenetics, defects of chromatin condensation and aneuploidy. So a proper feed to 1 carbon cycle has a potential to stimulate the endogenous antioxidant system defenses, that is glutathione synthesis, to activate the compensative homeostatic mechanisms and stimulate the body's own oxy-redox balance and DNA methylation, which are strictly cross-regulated. But the effect of dietary habits and supplements are variable because of individual genetic polymorphisms, individual environmental factors, and there are several enzymes which are affected. Oxygen metabolism is the main source of energy supply on this planet, and any healthy cell which is able to manage oxygen metabolism will prevent the damage from the oxidizing agents would survive. This damage by the oxidative species occurs when the cell body's own endogenous cellular antioxidant mechanisms are overwhelmed because of inflammation from inside or because of outside pollution or infection. The body's own endogenous antioxidant mechanisms are supplied by dietary substrates, micronutrients, and individual genetic substrates. These are the variables. And among cellular species, gametes, the oocytes in females and sperms in males, are most sensitive to the imbalances in the genetic problems because of the oxidative damage. Because they have a long maturation process, 90 days in case of 90 to 100 days in case of females, 70 to 90 days in case of males. The gametes have a specific task to collect the environmental signal and pass on them to the offspring in form of epigenetic marks, and these are always altered by environment and nutritional factors. I am again stressing the fact on environment and nutrition factors because they are the modifiable risk factors. The oxidative damage, it affects gametes, chromatin, and the genetics. Oxidative damage occurs in all the cells, but it is likely to be repaired. There is a huge but finite DNA repair capacity. The chromosomes of gamete, they provide a structural framework that is required for a proper embryonic development. But oxidative aggression affects the chromatin structure and the epigenetic regulation of gametes in two ways. There are two ways: epigenetic regulation and the chromatin structure. In sperms, they affect the protamine content, and in both sperms and ovaries, they affect the methylation. The epigenetic marks in form of methylation. The methylation of DNA occurs at cytosine residues, the parts of cytosine phosphate guanosine repeats, called as CPGs. These CPGs, they aggregate in an area called as CPG islands, and the extensive methylation of CPG islands causes loss of affinity of gene promoters for transcription factors and results in gene slicing, gene silencing. In addition to the methylation of CPGs, together with the histone lysine methylation, it regulates the chromatin state, transcriptionally silent state, that is essential for the maturation of both sperms and oocytes. These CPGs and CPG islands are the main target of oxidative damage. The oxidation of methylated cytosines with CPGs causes hydroxymethylcytosine, which is a starting step of demethylation and may result in the loss of epigenetic marks. The oxidation of methyl cytosine or guanosine to form 8-oxoguanosine within the otherwise properly methylated CPGs causes loss of inhibition of binding of transcription factors and loss of epigenetic regulation. This has been proposed as the main mechanism of loss of epigenetic regulation from the environmental damage leading to the neoplastic and neurodegenerative diseases, and is likely as a consequence of the excessive oxidant damage. The loss of epigenetic regulation in gametes leads to the erratic phenotype and decrease in the fertilization potential, possibly aneuploidy. Second mechanism is the chromatin structure. Oxidative stress-induced perturbations of DNA methylation also affects the chromosome stability and segregation and leads to the formation of aneuploid of both sperms and oocytes. Meiosine requires full chromatin condensation to facilitate the process, which is called as a heterochromatin state, and only the areas intended for the homologous chromosome recombination. The recombination hotspots remain hypomethylated and decondensed to allow for the recombination process. This hypomethylation of CPG islands attracts the recombination machinery. In sperms, the abnormal axial chromatids condensation during meiosis after chromosomes segregation alters the chromosome segregation by interfering with both chromosomes, microtubule interactions, and sister chromatid separations. The oxidation of chromosome telomeres consist of guanosine-enriched TTA-G sequence, which are the 1000 base pairs long. Telomere triple G repeats are preferential targets of oxidative damage in most of the oocytes. While this damage may be repaired in fibroblast, but in oocytes, this telomerase deficiency are prone to aneuploidy. The oxidative damage also causes overriding of the spindle checkpoint, the safeguard mechanism that halts the anaphase onset until the meiotic spindle assembly, and thereby providing a further link between the oocyte aneuploidy and oxidative distress. In summary, oxidative damage is likely to be clinical relevance because of the epigenetic regulation derangement and perturbation of the gamete chromatin condensation phase, which does not leverage on the repair mechanisms. The supportive interventions, they affect at restoring both 1-carbon compounds and folic acid metabolism, which is aimed at targeting the availability of activated methyl groups for DNA methylation and proper reactivity by endogenous antioxidant system that will protect the epigenetic marks from misleading oxidation. So from a metabolic point of view, 1-carbon compound metabolism and folic acid metabolism are two sides of the same coin. Our body's physiologic oxidation and redox homeostasis. In physiology, any active reactive oxygen species is neutralized by an antioxidant cascade. This includes both enzymatic and non-enzymatic antioxidants in the body. Enzymatic are superoxide dismutase, glutathione peroxidase, catalase, while the non-enzymatic ones are vitamin C, ascorbic acid, tocopherol, the vitamin E, carotenoids, flavonoids. On top of this cascade, there is a redox buffering system which is based on the antioxidant, the tripeptide glutathione. Glutathione can directly scavenge the hydroxyl radicals and oxygen it is able to generate most of the cellular antioxidants, vitamin C and vitamin E. While this glutathione is abundant in mitochondria and cytosol and nuclei, it is able to release the glutathione to any molecules. And this glutathione biosynthesis is by transsulfuration pathway. It is formed from homocysteine. This homocysteine, it reacts with serine to form cystathione, and that binds to glutamate and glycine to form an active tripeptide. This pathway of glutathione synthesis is paramount importance because glutathione must be available on site. There are no dietary sources of glutathione storage. The oxy-redox homeostasis is based upon the glutathione bioavailability, regeneration and de novo synthesis, when, where and how long it is needed within the delicate and homeostatic mechanism. The starting point of glutathione biosynthesis is de novo is homocysteine, which in turn is end product of one carbon compound, and this is the pathway of life on the planet Earth. The main pathway for non-photosynthetic organisms to add carbon unit to the molecules, and its massive cycling is essential for cell growth and differentiation. Briefly, the methyl group of methionine, and after an activating adenylation to form as adenosyl methionine, is released to any acceptor molecules and generates homocysteine. This homocysteine can further be remethylated to methionine with a methyl group donated from either methyl tetrahydrofolate, that is active form of folic acid, or betaine, which is a trimethylglycine, which is formed from the choline oxidation of mitochondria. The lack of homocysteine recycling and its accumulation is toxic to cells. It is a powerful antioxidant and it inhibits most of the fertility. Whether or not homocysteine will get converted to glutathione synthesis depends upon a key enzyme which is called as cystathione beta synthase. For its activity, vitamin D is required. For its activity, vitamin B6 pyridoxal phosphate is required. This cystathione beta synthesis is a tetrameric enzyme which requires vitamin B6 as a cofactor and also requires zinc in a chelated form. It is highly active. It responds to a very peculiar mechanism of action. This enzyme is the epicenter of cellular metabolism. This enzyme is made up of 4 CBS domains, in which heme is a central moiety. Iron acts as a redox sensor. In inactive state, it is ferrous, while when it becomes active, it becomes ferric. As iron oxidizes, CBS enters into primary active form, configuring a typical redox regulation that opens or closes by the transsulfuration pathway in a reversible switch. However, in order to become highly active, there needs to be a second activation of CBS by a methylation pathway. Transmethylation by a methyl donor generated from one carbon compound must bind to each CBS. That means 4 methyl should bind to 4 domains of CBS, which leads to an allosteric upregulation of enzyme, which increases the activity by 5-fold. This regulation of CBS activity is the core of cell resistance to the oxidative loads and is paradigmatic of how the cell homeostasis works. This one carbon compound pathway of adding carbon or methyl units to the molecules helps in the growth and differentiation of various cells in the metabolic activity. Within the pathways which are fed by one carbon compounds, this DNA methylation has a priority. The ability of cell to defend against the oxidation and thereafter is relevant for the cells that are duly growing with a positive balance of activated methyl groups to switch on glutathione synthesis, and otherwise the cell will be designated to the apoptosis. The final effect of antioxidant defenses will be active and efficient only in cells that are metabolically healthy. Many companies come to us with cocktail of antioxidants which contain coenzyme Q, which contain L-carnitine, which contain melatonin, they contain vitamin C, they contain alpha-lipoic acid, they contain astaxanthin, they contain selenium in various doses, various forms. While some may be helpful, sometimes may be helpful, but the small effect of pregnancy rate because of this aggressive oral antioxidants is explained by forcing the cellular environment to a redox state in front of a delicate metabolism of gametes, which necessitates a perfect oxy-redox balance. Strong antioxidant cocktail, it causes excessive removal of oxygen-free radicals, which are essential for the sperm function. This may negatively affect the capacitation and acrosomes and may induce a reductive stress as a rebound effect. This is shown by clinically confirmed by worsening of sperm nuclear condensation and padlocking of protamines, and a negative outcome of sperm DNA fragmentation. Also, whether these cocktails They can affect, they have a finite number of antioxidants, they have a finite amount of antioxidants, and they can affect only finite number of chemical reactions. While they may be advantage for a one particular chemical reaction, they cannot affect all the antioxidant pathways. So the cocktails of antioxidants, according to me, they are not suitable because they do not achieve a perfect oxy-redox balance. They do not achieve an efficient DNA replication and protemination as required by the gametogenesis. While the harmful effect of oxidative stress on gametes is unquestionable, there is a need to prevent the oxidative stress. There is a need to counteract the oxidative aggression. But antioxidants may not be the answer. The answer may well lie in feeding and stimulating the body's own one carbon system and folic acid metabolism. Once understood the role of one carbon compounds in activation of physiological antioxidant response, if we give a robust support to the one carbon compound, that can result in an improved DNA methylation, it can sustain the DNA maturation, gametes maturation, it can induce an activation of an endogenous antioxidant system and thereby configuring a physiologic and a self-regulated antioxidant activity, which is devoid of any reductive stress caused by the iatrogenic or oral antioxidants. This one carbon compound metabolism is strictly dependent upon the dietary sources of methyl donors and other essential substances. If the deficiencies of the dietary sources of methyl donor or other essential substances, which I'll come to it slightly later, then the endogenous antioxidant defenses may be poor. This endogenous antioxidant defense may also be poor in cases it is overwhelmed, like anti-inflammation in the body, or from the outside sources of infection or inflammation or pollution. So the dietary sources which are important for stimulating this endogenous one carbon metabolism are folates, the active form of folic acid, the L-methylfolate, 5-tetrahydrofolate, which is an important source of homocysteine remethylation. Riboflavin, the vitamin B2, and niacin, the vitamin B3, are important for the activity of MTHFR enzyme. Vitamin B12 is necessary to pass the methyl group from folates to homocysteine. And finally, zinc. There are no sources of zinc in our body storage. Zinc is only absorbed as a chelated form, and it is very important for the function of MTHFR enzyme, MTRR enzyme, CBC, CBS enzyme, cystathione beta synthase enzyme, by the means of zinc fingers. If these are given in active form, they have a potential to saturate and stimulate the endogenous one carbon compounds, generate an excess of S-adenosylmethionine for the activation of cystathione beta synthase, and for an efficient glutathione stimulation and production at any point of time. Glutathione synthesis can further be augmented by a cysteine donor, that is N-acetylcysteine. Glutathione synthesis and requires cystathione beta synthase enzyme, which requires vitamin D, which requires vitamin B6 as a cofactor. Vitamin E is also an endogenous antioxidant, but it also plays a role of stabilizer. It prevents the cysteine oxidation. While this active form of L-methylfolate, active form of methylcobalamin, active form of pyridoxal phosphate, vitamin D, cysteine, betaine, it stimulates endogenous one carbon compounds. It leads to an antioxidant modulation, but it does not lead to antioxidant gain. This antioxidant gain does not increase the reductive stress in the body. The sperm DNA maturation index is improved in males who have got high homocysteine levels. The oxidative damage to the genes as a result of the excessive DNA fragmentation is prevented by the endogenous stimulation of one carbon compound. So in short, a tailored nutritional intervention to support a one carbon compound and glutathione synthesis may have a fundamental effect on the reproductive prognosis of couples resistant to IVF and unexplained infertility. IVF failure may well be a metabolic problem. So rather than looking into the macros, the hormone levels, it is important to look at the micros. It is important to go into the biochemistry, the molecules. A simple activated form of L-methylfolate, methylcobalamin, pyridoxal phosphate, vitamin D, along with zinc and acetylcysteine, vitamin E may do wonders for people with infertility. And almost 50% of people with unexplained infertility, unexplained IVF failure, unexplained IUI failure, unexplained recurrent pregnancy loss, or even with people with past history of problems during pregnancy, early pregnancy loss, late trimester pregnancy loss, increase in preeclampsia, blood pressure, increase in incidence of abruption. All these people may benefit from one carbon compound. This may also stimulate the endogenous antioxidant pathway of our body, and this may also have its root in the way we age. This was a very difficult video. I had to make a detailed study. But if even 5% of the people start following what I mentioned in this video, the purpose of making this video may well be served. Please like the video. Please share the video. Please subscribe to our channel. If you have any comments, write to us directly. Thank you. Bye-bye.